T-shirts are, we have, an arsenal of ridiculousness to print on our T-shirts coming. And your names will go on the back, and it's a good time. So, meiosis. Last class, we talked about mitosis. And remember that when we talked about mitosis, we said we had one parent cell that duplicated its chromosomes and then divided in half, so that the two daughter cells were identical to the one parent cell. That's what we said. And so the reason we're doing meiosis at the same time is because that process of division is identical. The difference is that meiosis divides twice. So in mitosis, we had one duplication of genetic information followed by one division, and that yielded two identical daughter cells. All right, so that's what we talked about for the last two classes. In meiosis, we go through one duplication, and that's followed by two divisions, and that results in 4 unique daughter cells. All right, well, why do we do this? I mean, just for funsies, or is there some kind of purpose? Well, of course there's a purpose. So, oh, a little too far up. Focus fail. You got this, buddy. Yeah, that's a little different, huh? All right. What's that? Chromosomes organized into what we call a karyotype. Karyotype, that's me. It's not, I got that off Google, but we could believe that it's me, right? So that's a karyotype. So all humans, unless they have some kind of odd chromosomal abnormality, have 46 chromosomes. Okay. You have 23 pairs, and each pair contains 2. Okay, so that's what you got. If you're a boy, you have an X and a Y, and if you're a girl, you have 2 X's. Where'd those come from? Okay, well, I got half of them from my mom, and I got half of them from my dad. Thanks, guys. What a sweet gift. And when we go back, and I talked to you guys last class about mitosis, what I said was, well, every single cell in my body has the same chromosomes. Those chromosomes are the result of the fertilization of egg and sperm that later became me. And the egg came from my mom, and it had 23 chromosomes, and the sperm came from my dad, and it also had 23 chromosomes. Okay? And then fertilization happens, everybody pairs up, and this is what you get out of that sweet gig. So, as we do this, it's useful for us to look at, I color-coded them. Okay? Pink ones are the ones I got from my mom, and the blue ones are the ones that I got from my dad. Okay? Great. We call them homologous chromosomes. Okay? Homologous chromosomes. And what that means is they carry the same general information, they carry the same genes, but the alleles are different that I got from mom versus the ones I got from dad. Okay, so my dad had gray hair his whole life. So on the hair color allele, for me, it has gray. My mom has brown hair. Okay, so at the same place, on the same chromosomes, homologous pair, is the information for hair color. But the one I got from my dad said she should have gray hair. And the one I got from my mom said she should have brown hair. And I ended up with gray hair that you have to color, right? Because what else do you do? And so that's how it all works out. Okay? But I got one from mom and one from dad. And that's the same for everything. Okay? One from mom, one from dad. And they carry, like I said, the same genes on the same places, but they have different alleles. They have mom's alleles versus dad's alleles. Okay, so now let's suppose that Mr. Rogers and I decide that we're going to have a baby, and in doing so, we each have to produce gametes. And so remember that gametes are cells that have half the number of chromosomes. So egg, sperm, pollen, spores, okay? Half the number of chromosomes. So why is that so important? It's so important because I got half my chromosomes from my mom, half my chromosomes from my dad, but in the end, I need to have the same total number of chromosomes as my parents. Like my parents had 46 chromosomes, and I have 46 chromosomes, right? And then any child I have should also have 46 chromosomes. So I only get to give half of this to my child, because then Mr. Rogers gets to also contribute half of his into the mix as well. Okay? So the process of having those chromosomes, cutting them in half, figuring out which of these gets contributed to an egg, is meiosis. Okay? Producing these cells. When the cells only contain half the chromosomes, we call them haploid. 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 Okay? So now we're going to look at some cartoon cells. So a diploid cell is one that has all the chromosomes like on the karyotype I just showed. Okay, diploid. So they were paired up in that picture. They were all paired up. So we call that 2N. 2N just represents that they are paired. Okay? Two complete sets of chromosomes, one from each parent. Human somatic cells, like the karyotype I just showed you, has a ploidy. Ploidy is just what this is. We call this the ploidy. Okay? 2N equals 46. Okay? So every single body cell I have is 2N equals 46. A haploid cell, so one that has half the number of chromosomes, has only one set, which is one of each pair, but you have to have, could be, one from mom or one from dad. So when I go through this process of producing eggs, which one of my chromosomes, right, so this came from my mom and my dad, my mom and my dad and my mom and my dad, right? Okay, which ones then I contribute is random. I have to give one of these, one of these, one of these, one of these, one of these. But it's not like all the pink ones stick together, and it's not like all the blue ones stick together. They get to randomly sort. Okay? You get to randomly sort. Okay? So when they do that, we end up with N, so unpaired chromosomes, and a total of 23 of them. But getting from diploid to haploid requires this process of meiosis. When we look at these cells have already gone through synthesis. So remember we talked about synthesis last time, and that's making a second copy of the DNA in a chromosome, okay, so that they're identical. Two identical copies. They're glued together by cohesions. That's what we talked about last time. We talked about with mitosis, what happens is when we separate, we break all those cohesions down the middle, and then these two go to the two sides of the cell. Okay? This is a cell that we're going to say is 2N equals 6. 2N equals 6. Why? Because these two are paired. These two are paired. These two are paired. Okay. So everybody's paired up. We have a total of 6 chromosomes, and they exist in pairs, 2N equals 6. So we call this a homologous pair. We might call it homologs. Would be another word for it. And in each one of these, we have 2 sister chromatids. That's, words from last time. Once it's gone through synthesis, and it looks like the X, at that point, they're 2 sister chromatids. Just as a preview to what we're going to talk about later, we also have, so within one homologous pair, this has 2 sister chromatids, and this one has 2 sister chromatids. These are called 2 non-sister chromatids of a homologous pair. Okay? Because these two have different information on them. Sister chromatids are exactly duplicates. These are non-sister chromatids of a homologous pair. Okay, that's a mouthful, but it's going to come up again in about 8 slides. Okay? Non-sister chromatids of a homologous pair. All right, so let's look in at meiosis. Most of the steps of meiosis correspond to steps in mitosis, but with one major difference, and it's one I've already highlighted for you. In mitosis, we have a single replication of the chromosome, so we go through synthesis, and that's followed by a single division of those chromosomes, so we end up with two daughter cells that are identical. Okay, that's what I said over here. In meiosis, we have a single duplication, so one round of synthesis, but we divide twice. We call that meiosis number 1 and meiosis number 2. I like it when we just call them the numbers, 1 and 2. What happens first? 2 happens second. The result at the end of this process is 4 daughter cells that actually have half the number of chromosomes of the parent cell. Okay, that's the goal. Because then, when those gametes fuse with gametes from another partner, you end up with an organism with the same number of chromosomes as their parents. So this is just a summary of meiosis. We're going to look at a big scale first. And then we'll get into the nitty-gritty here in just a second. Okay. Overview of meiosis. So, first, what's the ploidy of that cell? Think for a second, tell your neighbor. Does it have a pair? Yes. 2n equals? How many chromosomes? 2. 2n equals 2.
Why? Never mind.
It's all right. 46 has been the right answer every time up till now. Changing it up.
I'm just going to go with 46 all the time.
Every single time. 2n equals 2 up top. What's the ploidy of this one? 2n equals 2. They have duplicated, they have not divided. Okay? This is why I ask you these questions in lecture and then again on the test. Okay. So then, what's the ploidy of this one? Does it have a partner? No. N equals 1. N equals 1. N equals 1. Ah! Okay. 2n equals 2, 2n equals 2, 1n equals 1, 1n equals 1. All of this is 1 equals 1. It does not matter if they have gone through synthesis or not. It does not matter if there are sister chromatids. Okay, that does not influence the number of chromosomes. It matters if they're paired and then the total number of chromosomes present. That's what matters. Okay, when we did meiosis, we took our chromosomes that had the 2 sister chromatids and we split them up, right? That's what we ended up with. So this part down here is very similar to mitosis, right? Because we had duplicated chromosomes and in the end they were just the lines. That's what we had there. Okay? So we call this step, meiosis 1, the reductional division. Reductional. Because what was my ploidy here? 2n equals 2, and this it is 1n equals 1. So this step right here is the reductional division. Because that's when we're changing our ploidy. Yeah, makes sense? Okay. Reductional division is here, and then this one is very similar to mitosis, where all we're doing is separating those sister chromatids. A few important things to keep in mind. Those sister chromatids are really just 2 copies of the same chromosome and they're identical, but the homologous pairs, these guys down here, came from the 2 different parents. They have different versions of genes called alleles at corresponding loci. This loci word just means location. Right? At the same spot in the same chromosome is information that codes for the same thing. Slight variations that we call alleles, but hair color, weight, shoulder breadth, all that kind of stuff. Meiosis is going to reduce the chromosome count to half, 46 to 23 in humans. I showed you guys the hedgehog, I think 2 classes ago. He was super cute with a little black nose. He had 92 chromosomes, I think, right? So his haploid cells would have 46. Okay, 46 chromosomes. Which is random? Like, which of those 46 goes into each cell, like I was saying before, that part is random. Okay? It has to be one of each pair, but which one of each pair is random? So, drill this into your skull. This is a big deal. Meiosis 1 separates homologous chromosomes. Meiosis 2 separates sister chromatids. Okay? So, next. Remember that? Heme cast. Here we go. Prophase, metaphase, anaphase, telophase, condense, align, separate, pulse. We do this twice. Because remember I told you we duplicate once, and then we divide twice. So we have to go through this twice. So let's take a look. Prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase. Condense. So all of the things that are happening with prophase are happening in prophase 1. See how we have ones? Guess what's coming after the ones? Oh, there they are. So good. Okay, so in meiosis 1, we have prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? In prophase 1, we lose our nuclear membrane. We start to see the condensation. Something else major happens here that doesn't happen in mitosis. Our chromosomes find their homologous pair. Okay? The chromosomes find their homologous pair. And they get together. It's like square dancing, right? You guys do that in elementary school still?
Oh, no.
No?
Video.
Watch the square dance.
Yeah.
Oh, the square dancing video. Okay. Well, I went to elementary school in the Deep South, and there they like one of your units in gym class was square dancing, and I like to not. And so I used to use that analogy when I taught this until I realized that you guys have absolutely no frame of reference for square dancing. Oh, we got one. We've got. Did you live in the Deep South?
Yeah.
Yeah.
Not deep, but south enough where that was a requirement.
Yeah, where square dancing was in the curriculum.
No, it was a requirement. You failed gym if you didn't.
You couldn't like do-si-do, right? Yeah. Okay. Knees up, folks. This is real. This is real. So not here in Colorado, I don't think. So here they've joined elbows with their partner, and we're ready, right? So now here we have in prophase 1, our chromosomes have partnered up, okay, with their neighbor. And then when we go to metaphase, look how they're sticking with their partner. See that? So wait, maybe a little bit more culturally relevant to you is the elementary school song, "The ants go marching one by one, hurrah, hurrah. The ants go marching one by one, hurrah, hurrah." I don't know the next line. You've never heard that song? Oh, we'll find it later on YouTube. They all go falling down. Oh, yeah, there's a rainbow.
And then it's 2 by 2.
Oh, they all go marching down
She's
to the ground. Yes, it's real. Down to the ground to get out of the rain. Boom, boom, boom. The ants go marching 2 by 2. Hurrah, hurrah. Okay, excellent. There it is. So now that we've found something that is perhaps more relevant than square dancing, the ants go marching 2 by 2. Okay, they have their partners who go marching 2 by 2. So then we attach our mitotic spindle, right? The whole 9 yards, all the things we learned about in mitosis, and then the chromosomes pull apart. Except we're not separating sister chromatids. We're separating homologous chromosomes. For the moment, we're going to ignore how come suddenly they're technicolor. We'll get to that later. What happened? But then, so, condense, cast, condense, align, separate, pulls. Condense, align, separated, pulls. See how nicely that works? We're going to reform our nucleus. We're going to go through cytokinesis in the same way we did in mitosis. And now here we are. We don't ever really uncondense at the end of meiosis 1. We're still condensed as we go into meiosis 2. But we did reform that nuclear membrane. It's kind of necessary to have that nuclear membrane in there before cytokinesis, for fear of something getting stuck in the wrong cell. So we have our nucleus that's reformed. And so in prophase 2, we again have to dissolve the nuclear membrane. Now we're lining up, and now those ants are one by one. You see them there? The ants are marching one by one. So this looks literally the exact same thing as what we saw in mitosis. Because now we've aligned on the metaphase plate, and as they separate, look at what we have. We just have one sister chromatid. We get to the poles, we reform our nuclei, and now we have 4 daughter cells that are all unique from one another. Why are they unique from one another? Because, look, here's the 2 red ones, the 2 blue ones. The blue one and the red one. Because the pinks don't all stick together, the blues don't all stick together, they move around, right? They get to assort independently. Do you want to watch the movie? Yes. Okay. It's like 4 minutes. Crotch zoom.
Focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Aww. Okay. There you have it, meiosis. So, I want to talk about that whole technicolor chromosome thing, like how come they were suddenly half red and half blue. We call that crossing over. Okay, crossing over. Crossing over is when we exchange homologous portions of non-sister chromatids. Okay, so in essence, go back to this thing. In essence here, I drew it up here. So if these are the chromosomes that I got, this one from my mom and this one from my dad, through crossing over, the two bottom halves would break off and they'd change places. Now you can't really tell on here that these are duplicated because once they're duplicated, they are glued together like they were showing in the video. So I drew them as far as X's at the bottom. So in essence, this is the chromosome I would have gotten from my dad. This is the chromosome I would have gotten from my mom. And then after crossing over, this is what I would be left with. Okay? So we're truly swapping portions. We break that phosphodiester bond. Okay? It's a covalent bond to the backbone, and there's a protein that goes in and breaks the bond and then reforms it in the other direction. That's what's happening during crossing over. So even so, what you're ending up with is that we have these 4 non-identical daughter cells, and the reason they're not identical, first of all, is because one of them's getting each member of the pair, but then second of all, because we don't even have 2 sides the same anymore, because one of these chromatids has exchanged information with one of these. So the reason we end up with 4 unique daughter cells is because of crossing over, and that's what this next slide is going to show you. So after crossing over, all chromosomes are this combination of maternal and paternal genes. So if I were to have a child, no matter what chromosomes went into my egg, both my mom and dad would have genes involved in that egg. Right? That's how you're inheriting your genetic information from your ancestors. Okay? So at the beginning of meiosis, those chromosomes are entirely maternal or paternal, but then after crossing over, they're a combination of those genes. If it weren't for crossing over, what we would end up with is, I'm going to show you. If it weren't for crossing over, these 2 would be the same, and these 2 would be the same. Okay, we'd end up with 4 daughter cells still, but there would only be 2 different ones. But because of crossing over, we end up with 4 totally different daughter cells at the end of this process. So this is a really good slide. I just put it in your notes so that you're aware that it exists, comparing mitosis with meiosis in the different places along the way. The place where this crossing over occurs is called a chiasma or chiasmata. Okay, that's the location where those bonds break and then they trade spots. Okay. Genetic variation produced in sexual life cycles contributes to evolution. So I would be remiss if I didn't point out to you the relationship between this and just how special you are. So mutations are indeed the source of genetic diversity for all organisms. So if you don't mutate, or I have to turn these back on. Sorry, video people. There we go. Okay, so mutations are where alleles come from. Okay, in essence, we talk about having an allele for brown hair, an allele for blonde hair, an allele for red hair. Where do those things come from? They're mutations that weren't bad. So they stick around in our genome, and that is where life's genetic diversity really comes from. Changes in your DNA create alleles. Well, reshuffling these alleles during sexual reproduction produces variation that results in a unique combination of traits in the population. So where does all of this variation come from? Well, it comes from the behavior of chromosomes during meiosis and fertilization that's responsible for this variation. This is the content of tonight's quad. Let me make sure. Yes. So 3 mechanisms contribute to genetic variation from sexual reproduction. 1, independent assortment, 2, crossing over, and 3, random fertilization. We're going to look at each of these in turn. We've already really talked about number 1 and number 2. So independent assortment was just saying, when I go through, all right, here's my karyotype. When I go through and produce eggs, the pink ones don't all stick together and the blue ones don't all stick together. We just get one of each pair, and which one gets contributed is totally random. It's a flip of a coin. Okay, so that is what we call independent assortment. Each one of those homologous pairs assorts independently of everybody else. And so what does that mean? If we're only looking at 2 pairs, what's the ploidy of that cell? Tell me. 2n equals 4. 1, 2, 3, 4. 2n equals 4. What's the ploidy of that cell?
2n equals 2.
N equals 2. Are they paired? No. What's the point of this one? N equals 2. They're the same. Okay? They're the same. All right. So if all the blue ones and all the red ones stuck together, you wouldn't end up with a whole lot of variety of gametes. But as it is, because they independently assort, you can end up with a lot of different numbers of gametes. And we're actually going to do the math here in just a second. Crossing over means that no matter what's happening with independent assortment, no one chromosome is entirely maternal or paternal. In humans, you have anywhere from 1 to 3 different crossover events per chromosome pair. This is a really common thing. Really, really, really common thing. Random fertilization. Okay. This is, like I said, this is your quad tonight. Sometimes it's a test question. I have no idea if it is this year or not. But every time somebody tells me random fertilization means that mating is random. And I think that that's left over from your experience with Hardy Weinberg, where random mating was a real thing. Anyone ever tells you that who you mate with is random, you should turn and walk away. Mating is not random. Okay? Never is that true. So, when we talk about random fertilization, it doesn't mean that who you mate with is random. What it means is that you, because you have 23 pairs of chromosomes, pair is 2, there are 2 to the 23rd different possible gametes you can produce. That's 8.4 million different gametes. Every one of us is capable of producing 8.4 million different gametes. I cannot crawl in my ovary and pick one and declare it as the egg to be fertilized. That's not how it works, right? That is random. Which one of those 8.4 million gets used is random. That's what random fertilization is. Okay, I don't get to pick that. Similarly, every male and female, so Mr. Rogers is also capable of producing 8.4 million different gametes. So he doesn't get to pick either. So when we talk about how many different unique children he and I can produce, that number is 70 trillion. 70 trillion different children that could be produced from one couple. And that does not include crossing over. That's simply off of independent assortment. There's 70 trillion. So when we talk about random fertilization, it has nothing to do with mate choice. It has everything to do with the fact that you can't pick the gamete that's used. They're all produced. Okay? There's no control over that. Yay! Okay, so